Welcome back to lab class. Today, what we're going to be covering mainly is the brain here, and we're going to jump into the plexuses as well as the meninges of the brain afterwards. We'll put it all together that way. So we're going to start off on page 26 on your checklist. Divisions of the brain is where we're going to begin. So I'm going to basically sit up a little closer. That way you can kind of see where I'm pointing and see the brain structure, just so you know what we're looking at. So we have the rear, the posterior. We have the most anterior portion of the brain, right and left sides, right? So consider like the brain sitting up in your head. We got a skull, for instance, here. See how it lines up pretty nicely right there. Good? All right. So we had some terms we had learned with the bones earlier. We're going to be going with some of those when we get to the lobes. Let's start with the basics, though. So letter A, cerebrum. Cerebrum is the entire wormy-looking portion of the brain here that we're seeing, okay? Cerebrum. So this is our general term, is what we could say here to begin with. We don't want to go necessarily beyond that for now. Other than that, we have two hemispheres. So cerebral hemispheres, if I go ahead and split the brain in half, okay, and we'll know later on that these two aren't, what we'll see is these two aren't connected actually. There's going to be a space here. But we have both a right and a left hemisphere, okay? And that separation point is what's known as the longitudinal fissure that you see in your checklist as the next structure. So if I take my pencil here, this line, the separation point, when I put the two halves together, when the cerebral hemispheres are together, this space, this line, that is your longitudinal fissure. Okay. Now the terms gyrus and sulcus, okay, let's go ahead and define those. Gyrus, or gyri for plural, okay, these are the folds of the cerebrum. You might want to say they're like the ridges, the portions that are sticking up. So if I just take my probe here and I go ahead and line up against one here, there we go. There's a gyrus. Here's another gyrus, right? Here's a gyrus here, gyrus here, gyrus here, gyrus here. All the wormy looking structures that are sticking up. Right? Now the sulci, plural, right? These are the spaces between the gyri. These, I like to call them shallow grooves. That's the easiest way to take a note, shallow grooves. So where we see the separations between the gyri, gyruses, gyri, right there, that groove that I'm poking the probe within, this is a sulcus. Now we're going to need to know a couple specific ones, okay? But we'll get to that here in a little bit once we separate the lobes a little bit more too. All right, so brain in general, right? We see cerebrum. That is the majority of what we're seeing. There's going to be 5 lobes that we need to know for our class. First off, we have the frontal lobe. That's pretty easy to suggest, right? Frontal bone, frontal lobe would be on the most anterior portion. So we're looking at frontal lobe as being this portion right through here. We're going to learn here in a little bit that there's these 3 major sulci, and these are what's going to separate right here. This sulci right here is going to separate the rest. So we know that anterior for now, frontal lobe. Okay. Next portion, parietal lobe. We had found that later on earlier too, right? Upper and more lateral portion of the skull. So we have upper and more lateral portion of the brain. If I take my skull here side by side, right? Where do we see the frontal bone? Frontal bone here, frontal lobe here. We see parietal bone here, parietal lobe here. So same locations, nothing's going to change face to face. Temporal. We had temporal bone here. Remember that squamous suture from before? Squamous suture? Well, simply enough, we go below this little line for now, we're going to call it, right? Just a generalized sulci, or sulcus. But we have temporal lobe right through here. We go and flip posteriorly, right? We had occipital bone. Now we see occipital lobe here, the most posterior. Last but not least is insula, or island lobe. To find that, I'm going to give you a whole different model because this is an in-depth cut. So if I take my, hello, I can figure out how to put this cerebrum, this left hemisphere back together, we'd be cooking. So we can see the same structures: frontal lobe, temporal lobe, parietal lobe, occipital lobe. Now we're going to go inside, right? Cool, we can pull that apart and then we can look. So what do you see here? You see this protrusion. Let me turn it around that way. This is called the insula, the island lobe. It's called insula for the test, but it's the only deep lobe we're going to ask you for. Deep to temporal. Page 27. Some basics that we want you to know, okay? The first structure, it says cerebral cortex. For cerebral cortex, then it will be the outer portion of the cerebrum, okay, outer portion of the cerebrum. So if we want to label that as the outer portion, we're simply just going to say, oh, this outer edge here. Nothing deep. Cerebral cortex, the portion, or you could say the outer portion of the cerebrum. And the cool thing about this outer portion, it's going to help us making new thoughts. It's going to help us make new decisions. Pretty cool portion about our brain. Not that you need to know that for lab portion. Moving forward, though, let's see where I'm at your checklist. Gray versus white matter. Now, this is a great model to show you that cortex, like I was talking about. You know, you can still see the outer gray per se compared to the white. Well, now we're going to flip that a little bit more. If I'm saying name this specific region, yes, we're looking for cerebral cortex. If I said name this specific lobe, right? Frontal lobe. Different kind of terminology, so to speak. Now we might ask, what kind of matter is this made out of? Gray versus white matter. And all we want you to know is that the outer portion of the cerebrum is made up of gray matter. So in this model, it shows it perfectly. Anywhere you see the wormy gray aspect coming around, that is your gray matter. All right, gray matter. And then where we see the white here, white matter. That's all we need you to know for now when it comes to lab material. And this would be the model we would use too. Next portion. Okay, notable fissures, gyri, and sulci. This will help you to see those lobes a little bit clearer. Sometimes I like that the checklist had this before, but we've changed it since then. But we'll go back to our entire cerebrum that we're looking at with the brain. The first portions that we want to look at under number 9, letter A, is the lateral cerebral fissure. The lateral cerebral fissure, okay, simply, right, lateral cerebral fissure. One large line along the lateral portion, or one, I should say, large sulcus. Now, we have that in parentheses, lateral sulcus. Go ahead and use that. It's simple if you want to call it. But lateral cerebral fissure is absolutely acceptable then as well. So that's what's really separating our temporal lobe from the parietal and our frontal lobes then too. The lateral cerebral fissure. The parieto-occipital fissure, on the other hand, you're going to have to go ahead and split the hemispheres apart. And what we're going to look for is one definite, okay, fissure, or again a sulcus. So you see this one right here, runs all the way up, all the way up to the cortex. This is going to separate the occipital lobe from the parietal lobe, hence the name parieto-occipital fissure. So it's one complete sulcus that runs all the way out to the cortex. That's the key factor there that tells us that that's the correct fissure we're looking at, parieto-occipital fissure. Now we're going to put these together. But I'm only going to use one side. I'm going to go ahead and use this right side. Now we're going to look for 3 major sulci. And these sulci are going to be called the central sulcus, the precentral, and postcentral sulcus. But we have to find 3 major grooves, right, that are going to denote these features. So we're going to look from the lateral fissure here. We're going to look for 3 grooves that run all the way up then to the longitudinal fissure. And if I'm looking at the longitudinal fissure right here, I can see 1 groove, 2 grooves, and 3 grooves. Make their way all the way across down to that lateral sulcus. So if I take my probe and I go right down the middle line of all of the 3, right? We got 1, 2, and 3, all right? I have found my central sulcus. Central sulcus, oh, hello. Central sulcus, all the way up to longitudinal fissure. So the central sulcus is our key feature when we're looking to separate the parietal lobe from the frontal lobe. Okay. Now we have a couple more we want to denote. If we find the central sulcus, we can move up to what is known as the precentral sulcus. And again, runs from the longitudinal fissure all the way down to the lateral fissure. Precentral sulcus. Move back to our central sulcus, and we have another one, right? The post-central sulcus. Runs all the way up again to the longitudinal fissure, all the way down to the lateral sulcus. The three major sulci that we need to know. Now, I did skip a couple things, precentral gyrus and postcentral gyrus. So if you find the central sulcus, you look forward and backward one. So again, my probe's on the midline, the central sulcus. If I move forward to this gyrus, okay, this is the precentral gyrus. So if I said name this fissure, we're looking for precentral gyrus. Name this fissure, or not fissure, I should say, but gyrus, postcentral gyrus. So precentral gyrus, postcentral gyrus, separated by the central sulcus, precentral sulcus, postcentral sulcus. There we go. See them wrap all the way around. So look for those features, those are key. Flip it around. Same thing, another numbered. We can see the central sulcus right there, numbered, with the precentral gyrus, postcentral gyrus, precentral sulcus, postcentral sulcus. Let's go ahead and split the brain. Mid-sagittal cut. There we go. All right, so now we can see the innards. We're going to go with number 10, corpus callosum. That's that big humongous C shape you see right here, kind of in white. Corpus callosum. Corpus callosum. Okay. So the cool thing about the corpus callosum is that is the meeting point of the more central portion of the brain here. As we said before, the two cerebral hemispheres are split, but then the brain does merge back together here at the corpus callosum. Corpus callosum. The fornix, okay, is the next structure. The fornix, simply enough, okay, what we're going to see that it's just slightly inferior. The fornix is this little sliver. A small little sliver that we see just below the corpus callosum. So if we look at the fornix and we look at the corpus callosum, there's one little pocket here. I can stick my probe right in that pocket. And that pocket is known as the septum pellucidum. Right now it's not punctured, but if we were to puncture the septum pellucidum, it would allow us to stick our finger, in this case, into the open spaces known as the ventricles of the brain. We're not going to get to those immediately here in a little while, but just so you know, there is, yes, open space. So yes, I have a hole in my head, definitely. There is one hole, if I were to puncture here, up into the right side, up a hole into the left side of the brain. But for now, septum pellucidum, that pocket there, separated by the corpus callosum, and then that little sliver there, the fornix, and that the same hue, same tan. Let's get into the functional areas of the brain now, just to take a little break from this model. And when we're looking at the functional areas, we're going to give you a color brain model to be definitive on these. So if you can kind of memorize the colors and the locations and their basic functions, you should be absolutely fine with this portion. Number 13, letter A, primary motor cortex. The primary motor cortex is also known as the precentral gyrus. So if we simply just look at this central sulcus right here, let me turn here, right through here is the central sulcus. The precentral gyrus is labeled in red here, okay? But for our purpose, when we're talking about functional areas of the brain, the functional area of the brain, the name of this portion, in red again, will be known as the primary motor cortex. The primary sensory cortex, we're just going to look posterior. The primary sensory cortex is going to be seen as light blue, and that is the post-central sulcus. So we had the central sulcus here, right? Central sulcus. So we move back to the post-central sulcus. We can see that there. So this space, this gyrus in between, this post-central gyrus, this is known as the primary sensory cortex. Light blue on the model. Take that note, light blue. primary sensory cortex. Motor speech cortex, we like to call this one the Broca's area in class here. So the Broca's area. The Broca's area, the color is going to be orange on the model. So we're looking just lateral and anterior to that precentral, or I should say the primary motor cortex here for now, functional area. So in orange is the Broca's area, the speech center of the brain, motor speech, formulate, actually say those words. The next portion, it says the sensory speech area, Wernicke's area, okay, another one that we really like. We just move posterior to the Broca's area to this weird brownish color, okay? So it's lateral and slightly posterior to the primary sensory cortex here. But this grayish brown area right through here, Wernicke's area, the sensory speech area. Next one on your checklist, auditory cortex. Well, we hope that's closer to the ears, so in the temporal region we're going to find that here. So if we find that lateral sulcus, we move just lateral and inferior. See this dark green color, this really dark green color? The dark green color, that is going to be the auditory cortex found within the temporal lobe. I skipped one, visual cortex. Well, you think the eyes are here, right? Kinda, absolutely, you could say. But the eyes have to go somewhere. The visual cortex is going to be within the occipital lobe, and it's going to be the most royal blue color. This is where the information is actually being interpreted in our brain, so we can see it. It is, yes, received, more anteriorly, we could say, but we need the eyes. We don't see any eyes here, right? I don't see any eyes. Those have been plucked out. But in order for us to interpret that information, we're going to need what is known as the visual cortex in blue. So again, occipital lobe, most posterior, royal blue color. Letter G, olfactory cortex. Sniffers, right? You smell. Well, there we go. Almost looks like little olfactory cortex. Looks kind of like little pointers coming off the brain right here. So here we go, olfactory cortex. We can see it extending through here in light blue. If we split it apart, you can see an underside right through here then as well. This little portion right through here, light blue. Right through here, though, especially light blue too. Olfactory cortex, the light blue on the underside of the brain. We'll come back to cerebral nuclei. You can go into your APR and find that one. APR for the nervous system, go to the brain. Cerebral nuclei, we're going to find those deep shadows that are going to be seen near the ventricles when we're showing you pictures. So look at those pictures on APR for that information, cerebral nuclei or basal nuclei. Letter 14, okay, or number 14, hello. We're going to move down, so inferior view of the brain, structures on the inferior side. I'm going to pull this model back out because I like it. Works well. Show you all these structures. We're going to go one at a time here. So you can see the inferior portion of the brain pretty well. We already labeled some portions, so we had talked about the olfactory portions, olfactory cortex being more deep. We can see there, again, olfactory cortex, light blue right through there. Well, we have to pick up that information from the more anterior portions of our nose. And that's where we're going to start here. The olfactory bulb. Simply enough, the olfactory bulb is this most anterior portion, just the round portion through here, olfactory bulbs. If we follow those olfactory bulbs posteriorly, we have olfactory tracts. Olfactory tracts. The optic nerve, we're going to see that this has been severed, right? Anytime you pull the brain out of a skull, the eyes are going to stay in place because there's only a little hole for the nerve to run through. So we are always going to see on brain models the optic nerve. This has always been cut. But we can keep following it posteriorly. So if I move sideways a little bit, see this little bean structure, we'll talk about that in a little bit. But we can kind of see a meeting point or an X point of these optic nerves. And that little X point where we see the two optic nerves converge is called the optic chiasm. They crisscross across one another, and we can see the latter portion of the X. And these are going to be called the optic tracts. So let's go through that line one more time. Olfactory bulb, olfactory tract. We can see optic nerve. Optic chiasm is the center point, and moving backward, optic tracts. Letter F, pituitary gland. Well, that's that little bean I was telling you about that was hanging there. It produces a lot of growth hormone. Keep our body more young, supple, smooth, muscular, burning fat, healthy. Good little thing right there. That sits in the brain, right? On the posterior, not posterior, but inferior portion. So generally speaking, if you take a really good look, I'm going to pull this apart. If we do a lateral mid-sagittal view, right, we can still see the pituitary gland, and it's hanging by a structure. And that structure that's hanging from is called the infundibulum. The infundibulum is this elongated structure which holds the pituitary gland in place. We can still see some of those inferior structures, right? So we have right there the optic nerve, which has been cut. We can see it move back to the optic chiasm. And then so just posterior to the optic chiasm, we have the infundibulum with the pituitary gland hanging off of it. Mammillary bodies, too. Let me put this back together. Mammillary bodies are going to be found just posterior to the infundibulum. They kind of look like a couple of bulbs, you could say. I'll put my probe on it right there. There's a bulb there, and there's a bulb just lateral to it too, two bulbs. So a bulb on either side, just posterior to the infundibulum. Mammillary bodies. Let me show you that mammillary body then from a lateral view too. There we go. Mammillary body looks like a little bulb right there. Other side, we got the infundibulum, right, with the pituitary gland on it, right there. Mammillary body. Trigeminal nerve, we can't really show you it too well, but we can see a collection of nerves running off along the brainstem here. APR again, there's a wonderful picture that shows you a trigeminal nerve. It almost looks like it's wrapping around, and down and around three major points, seen originating along the brainstem. Trigeminal nerve, so APR, that one, very good idea. Arterial circle, we showed you that before. That was with the skull model. There was an awesome picture on APR, but again, we can see that collection of blood vessels right through there. All right, so you can see those collection of blood vessels. The circle of Willis surrounds where the pituitary gland would sit. And they'll sit in the sella turcica. See if I remember that from last unit. Let's keep moving down though. Brainstem. Okay, we're gonna go ahead and split this apart again. No, actually, yeah, let's stay. Brainstem. Now with the brainstem, okay, there's some major things we want to know about, and we need to be able to define the separate portions of the brainstem. The brainstem, the first portion that we want to look at is the midbrain, and I'll come back to some of these other structures with the diencephalon after. But the brainstem, or midbrain, is going to be this most lower portion, we could say right here, posterior to those mammillary bodies, and it's going to run straight downward towards what is known as the spinal cord, right, at the very end. But we need to break these down. We need to know what each of these separate portions are called. Cerebral peduncles are the first ones. I like to call those the pedestal. So this portion right through here is the cerebral peduncle. But if we look at it from the underside, it's a little difficult to see per se. I'm going to take my probe in here, and I'm going to, there we go, stick my probe on it. So it doesn't really show it too clear, but that entire portion that I was sticking my probe on lines up perfectly in place with this portion, the cerebral peduncle. I'm going to grab my brainstem model so I can show you that again too. The next thing I want to show you is the superior and inferior colliculi. So we have these two little bulbous looking structures here and here, all right? So we find the most posterior portion of the corpus callosum, move inferior to it, just posterior to the cerebral peduncle region. We have one little bulb here, superior colliculus, another little bulb here, inferior colliculus. Together they are the colliculi, and if they are bilateral, we have superior colliculi, inferior colliculi. The pons, looks like this little turkey gobbler right here. The pons, the bulbous looking portion of the structure. The pons. And if we move down from the pons, we come to what is known as medulla oblongata. Vital reflex center is found here. So when somebody tickles the bottom of your foot, that little reaction of the curling of your toes, right there is where that's coming from. Always works, no matter what, even if your brain dead, for the most part. Let's move back up to letter B, the diencephalon region. So we had found the midbrain, in general, midbrain, cerebral peduncle, all the way down to the medulla oblongata. So we've moved to this more central portion through here. This is the diencephalon in general, diencephalon. Let's break that down a little bit more. Thalamus versus the hypothalamus. Simply enough, we look at where this almost looks like an eyeball structure is. And this little space right here, spot right here, is the thalamus region. If we move down from the thalamus and slightly anterior, we have this little portion through here. So this was the optic chiasm, right? We move up from the optic chiasm and we find the hypothalamus. So another structure we can see from the side here, from this mid-sagittal cut. Again, optic chiasm. Then we see the infundibulum essentially hanging from the hypothalamus. And then we see the mammillary body again one more time, all in a row. Back to another diencephalon structure. Towards the most posterior aspect is going to be the pineal gland or pineal body. So I'm just going to go ahead and take my probe and stick it on right there, that little bulbous portion that we see right through here. Pineal gland or pineal body. We'll take either or, absolutely fine. That's going to be helping us with a lot of our sleep aspects. Learn about that more in the lecture. Keep on moving down. We had already touched on those structures, right? Mammillary body again, infundibulum, and optic chiasm. So we should be really, really well on those, whether we're looking at that from an inferior or a mid-sagittal cut. Letter D, cerebellum. Cerebellum is this more, I guess you say, brown-hued portion that sits posterior and inferior to the cerebrum. So cerebrum, this bulb here, cerebellum. From the cerebellum, we need to know two portions of it. You can see the folds along it. Those are called the folia. Folia are the folds on the outside of the cerebellum. And then last but not least, we pull it apart. We look at the cerebellum. We look at this tree-looking structure in white. And that tree-looking structure is arbor vitae, translated tree of life. Arbor vitae. Okay, I'm going to pause and come back to your minute. Throw this in here real quick. Here's your brainstem model. Looking at that, we can see portions of the diencephalon, the insula, as well as the brainstem itself. So we have the midbrain, right? Midbrain comes through here. Diencephalon is going to be found deep, but we can see the insula here. This is the insulary lobe. If we move to the most posterior aspect, okay, you can see some definite structures that we were looking at before. This little pink little tongue here, that's the pineal gland or pineal body. And just lateral and posterior to those, we have the superior colliculi, followed by the inferior colliculi. Excellent choice for a model with that. We go into an anterior view, right? You can see an optic nerve's been cut, but we could see the optic chiasm, the meeting point, the optic tract, moving posterior right through there. We can see those two little white bulbs in there, mammillary bodies. Infundibulum looks like it's been busted, but we can kind of see a remnants of that then too. We can definitely see the cerebral peduncle of the midbrain. Cerebral peduncle looks like a pedestal. Just below it is going to be the pons, and then where my fingers are holding right there. There you got your medulla oblongata again. pull this apart, give you a internal view. So again, medulla oblongata, pons, cerebral peduncle. You can see a mammillary body, just anterior there in white. You can see those superior and inferior colliculus sitting just above in pink, pineal body or pineal gland. Now we can see the thalamus again one more time with the hypothalamus here. And what did we see right there? Optic chiasm one more time. This space right here, what we'll come to find out, is just past that corpus callosum through that septum pellucidum, and that's opening up to the space of the lateral ventricle. We'll get to in a little bit. Here we go with the ventricle model. Okay, you can see this one in gray. Note that where every gray is as a structure for this model is actually technically empty space, open space where cerebral spinal fluid sits, right? So cerebral spinal fluid is going to be found in this gray portion. So if we line this up, right, we can kind of see with the brainstem model how it would fit ever so nicely that this space right here on both sides would be open. Fluid is running through these portions. You can see where that little eye was, right? Remember that eye where the thalamus sits? Where do we see a hole? Right through there. So that hole is now a real space here. Get it? Kinda? That gray portion that we see around the hole is this open little pocket where I can stick my finger right there. So let's break it all down. I'm going to give you as an anterior view looking at you. Here's your lateral view, left side. These two horns that we see, these two horns I'm going to put my hand on right here. These are known as the lateral ventricles. There are two, 1 and 2. Don't have to number them, just know that these are lateral ventricles together. Okay, from the lateral ventricles, we see them go through what are known as the foramen of Monro. It also says interventricular foramen if you want to use that. Great, interventricular ventricle between foramen. Okay, makes sense. But foramen of Monro, real easy to remember that way. That's going to be that tiny shaft right there that runs from the lateral ventricle down to what is known as this portion here, the third ventricle. So let's zoom in one more time. Lateral ventricle, you can see that big C shape. That portion right there that runs downward, foramen of Monro. Then this portion where I'm hitting with my probe and this big circle area, all through here, third ventricle. Foramen of Monro, third ventricle. From the third ventricle, we run downward and we see what is then known as the fourth, or sorry, not fourth ventricle, but the cerebral aqueduct. The cerebral aqueduct is going to run down posteriorly, okay? So let me stick my probe on that, right through there. There you go. There is the cerebral aqueduct. If we go forward, we get back to the third ventricle. If we go posteriorly, we see this one little portion hanging off the back, and that is the fourth ventricle, okay? So let's go through that one more time. You can see the lateral ventricles. You can see technically two foramen of Monro, one there and one there, connecting to the third ventricle. This entire portion right here. The cerebral aqueduct extends downward, and then we come to the fourth ventricle. Last portion you need to know is anywhere in pink where we see a ventricle, that is called choroid plexus, and choroid plexus is the pink area where cerebral spinal fluid is being manufactured.
The protective coverings of the brain and cerebral spinal fluid. Page 28 on your notes, letter A, meninges. So in general, we have meninges. We're going to break these down one by one with your checklist. Dura mater, in Latin, tough mother, is this outer portion that we're seeing along the entire brain. So it's the outer covering, and we will not see any of the gyri or sulci of the brain. So it's just a generalized coating, almost like you could say a helmet per se. And what you'll notice is it is a very thick and very strong structure, very tough. Tough mother is the name, so dura mater makes sense to call it dura mater. Okay. Now what we're going to notice, once we pull the dura mater away, we can see the actual brain. Now with the left cerebral hemisphere, okay, we can see a slight coating there, versus the opposite side, we don't see a coating. So what we're poking up with the probe, that is going to be the arachnoid. This actual thin portion we can pull up off the sulci and gyri. Now if we look to the opposite side, okay, the right cerebral hemisphere, we'll notice that that coating, the arachnoid, is gone. Now if we were to pull and pick away at the gyri, at the sulcuses, we're going to see that we're going to be tearing away what is known as pia mater. So it's super thin, tightly attached, we could say, with pia mater, just like with the spinal cord model, tightly attached. If we look along the midline, okay, that longitudinal fissure, we can see granules. These are known as the arachnoid granulations. Okay, we will only see those along the longitudinal fissure of the brain. The last structure that was in there was the cerebral sagittal sinus. If we go ahead and look at the dura mater, running along the dura mater, there is one opening, and that space is going to allow for cerebral spinal fluid to flow through. That will be our
Hold on real quick. Let's get
All right.
that mid-sagittal model.
Sure thing. That would be the superior sagittal sinus. So it's a good way to show you it here on the brain. Imagine that space going into the longitudinal fissure right through there. Because remember, the cerebral hemispheres are separated. Now we do have a model here. One more time, the cerebral. One more time, the superior sagittal sinus. We're going to see that within the dura mater as that open space there, and it's going to run along the midline. So right through that longitudinal fissure. But if we look at an actual model, okay, do you see where we see this open blue space? That is going to be the location of our superior sagittal sinus, all the way through. So that's another way we could test you on it. But since we're at this model, we can go ahead and show you some ventricles one more time. We can't see it, but through the septum pellucidum here, if we were to open up through there, we'd see the lateral ventricle. But if we just move down more inferior to it, we can imagine where we would see a space here for a third ventricle. But to connect the lateral ventricle to the third ventricle, we would have to have the foramen of Monro, which is tiny little open space that we see right there running upward. If we move down, we have the cerebral aqueduct. The cerebral aqueduct is going to run just posterior to the pons, just anterior to the cerebellum, and open up to what is known as the fourth ventricle right there. And those are your ventricles for a mid-sagittal view. These pictures can be found on APR. These are the plexuses models, or pictures. So what we're seeing here in the upper portion of the neck is going to be the cervical plexus, specifically. If we look down at the next picture, we've moved down to what's known as the brachial plexus, and we can see that extending out to the axillary region, so brachial plexus goes towards the arm. Flip our picture, we can go ahead and look at another version of that brachial plexus here. You can see what's known as the phrenic nerve on another picture here. We'll see two nerves running on either side past the heart here, which run down all the way to the respiratory diaphragm. Phrenic nerves stimulate that. Going deeper, we can see the lumbar plexus. The lumbar plexus we can see originating from the lumbar vertebra here, and they run down through the pelvic cavity. And then simply flipping the picture one more time, we drop down a little bit more, and we can see the sacral plexus. Structures we skipped over. When you go back to other structures associated with the spinal cord, right, and nerves, we're going to cover all those at once here.
Who is this?
I touched on the phrenic nerve already. So what we're touching on here is that trigeminal nerve that I told you about. Trigeminal nerve sits on the lateral portion of the face here, but we're going to see that connecting to the midbrain. But we can see how we have three major portions really coming off and around the brain itself. If we skip ahead to basal nuclei, I told you about those. We're going to find those just lateral to the ventricles. But we're going to be seeing those as these gray structures, but they're highlighted in orange on this picture for us to see the basal nuclei. If we move a little bit forward, I can show you another view of the insula there. So you can see the insulary lobe separation here. So you can see that's just lateral to those basal nuclei, just deep to the temporal lobe then as well. A few more structures to take on a field trip for. We take this model. Cool model, right? We can definitely see a spinal cord, right? No problem at all. Spinal cord, spinal cord, spinal cord. Now those other structures that we want to associate with the spinal cord, first off would be the cauda equina. The cauda equina can be found at the most tail end. Horse's tail is the translation. But cauda equina, the tail end of the spinal cord that splits down offwards towards the pelvic region. Dorsal root ganglia, just to show you those dorsal root ganglia, we're going to see those, we saw those before with the spinal cord model, but the dorsal root ganglia are going to be these little bulbs that we see here. And when we extend outward, that's what's going to be forming the spinal nerves we saw earlier on the spinal cord model. The last structures we need to know along with this model, all right, are these other bulbs that we see running parallel to the spinal cord here. So these other structures here, these little bulbous looking structures, are going to be sympathetic chain ganglia. Sympathetic chain ganglia. The final nerve on your checklist was that
sciatic nerve. And then if we come down from where the cauda equina would be originating through here, we see one large nerve shooting out through the greater sciatic notch of the ilium, and that would be the sciatic nerve, the thickest nerve that you can see there running through the entire posterior thigh.